WHO EPI-WIN Wednesday webinar of March 2023. For those of you who are new to EPI-WIN, it stands for the WHO Information Network for Epidemics, and it's a platform for us to get the science out to you through global experts and make the science accessible, understandable, and relevant for you. Today, we have a very important topic. It's on influenza vaccines, what makes them work. And moderating today is Dr. Wenqing Zhang, who needs no introduction in the world of influenza. She's the unit head for the Global Influenza Program in WHO, and she will be moderating it. And we will have the opening remarks by our director, Sylvie. So over to Wenqing now to moderate. Thank you very much.
Hello, everyone. Can you hear me? I think so. Great. So let's kick off this webinar. First, let's welcome our director, Dr. Sylvie Brown, to give the opening remarks. Dr. Brown, Sylvie, please.
Thank you very much, Wenqing, and thank you, Supriya, as well for organizing this EPI-WIN webinar. And welcome to all of you, and good morning, good afternoon, or good evening, wherever you are. And it's really a pleasure for me to be with you today, because I can tell you this is a fascinating topic. I think for me, it's one of the most fascinating topics of public health, is how we can develop the best vaccine possible to protect people against influenza, and especially the severe form of influenza. As you know, every year there are between 400,000 to 600,000 people dying from influenza, and mostly those people are elderly, people with underlying conditions like cancer and so on, and also pregnant women and very young kids. So the vaccine is a really important public health tool to protect those people and prevent them from having severe infection, being hospitalized, and for some of them dying. So, but this public health tool, this vaccine, has to be adapted because viruses are changing, and so we have to adapt the vaccine. So that's why I think the process by which we decide what vaccine will we have for next season is absolutely fascinating, and today we have been able to have two world experts on the topic, and so I hope you will really enjoy the conversation with them, and we are very much looking forward to their presentation, but also to the discussion that we can have after the presentation. My last point will be also about pandemic prevention. As you know, there will be other pandemic in the future, and the most likely origins of pandemic is some viruses from animals that spill over to humans and then create a pandemic situation. We have seen this with COVID-19 and with many other instances where it's an animal virus that spill over to human and then is amplified and create epidemics and pandemics. So it's very important because in case we have such event with avian flu virus, we need to be ready also to have a vaccine to protect people around the world. And that's why I think we will address those two issues today, not only the process by which we can produce a vaccine to protect people against seasonal influenza, the one that comes back every year, but also the process by which we can also anticipate potential emergence and preemptively start preparing vaccines in case such avian flu virus or swine flu virus becomes a pandemic virus. And this will help us to make this world a much safer place to live in. So with this, I leave you with those experts and Wenqing Zhang, who is unit head of the Global Influenza Program. She has years of experience in influenza, and she's really one of the most prominent figures in WHO on this topic. So Wenqing, back to you, and thank you very much to David and Richard for being with us today.
Thank you so much, Sylvie, for setting the scene. My role today actually is moderator, and you know this is very light role in this webinar. Just to say that this is one of a series of webinars we're organizing on the topic of influenza. Today, we are going to talk about influenza vaccines, but we'll be focusing on what's inside influenza vaccines. We have two topics. One is on the seasonal influenza vaccine composition, what's inside the seasonal vaccines. And the other topic is about avian influenza, including H5 and the candidate vaccine virus for zoonotic influenza for pandemic preparedness purposes. Without further ado, let's welcome our first speaker of this webinar, Dr. David Wentworth. So David is the director of the WHO Collaborating Center for Influenza in CDC Atlanta. He's also the chief of the Virology Surveillance and Diagnostic Branch in the Influenza Division of CDC. Dave, you have the floor.
Thank you very much, Wenqing. I'm going to go ahead and share my screen. I'm also going to turn my video off now so I have more bandwidth to ensure I have that. Hopefully you can see my screen at this time. Could somebody let me know if that's the case?
Yes, it's perfect.
Excellent. Okay, so we'll dive in. And what I'm going to tell you about today, I was asked to tell you a bit about how the vaccine strain selection process works. And by understanding that, you will get a good sense of the major overarching topic of today's webinar, which is, basically what makes them and what makes them work. And so we're going to talk a bit about background of the viruses to level set and get everybody up to speed on how flu viruses work, just very briefly, and the inherent challenges that they pose. And then we'll talk about the key questions addressed for vaccine antigen composition and the types of data that are needed to address those questions. And then a few examples thrown in during the course of it of how these questions specifically are addressed with scientific examples. And so that gets a little granular, but I hope to cover a wide breadth of how this process works. So influenza A and B viruses, these are viruses that normally infect humans. We have in our population now influenza A H3N2 viruses, influenza A H1N1 PDM09 viruses. And so these two viruses are actually both relatively recent introductions into our population. H3N2 came in the pandemic of 1968 and stayed with us since then. And H1N1 came in the pandemic of 2009. We previously had a different H1N1, and that's why it's called PDM09. So they're both pretty recent introductions into our population. The influenza B Victoria lineage viruses, and then the influenza B Yamagata lineage viruses. And something interesting has happened with them in that they have declined significantly and none have been detected since March 2020. We're still trying to determine now if they're truly extinct or not. So the virus is shown over here as a schematic with these blue peplimers sticking out of the top. Those are the hemagglutinin proteins, and these red surface proteins here that have 4 parts. So the hemagglutinin has 3 parts. It's called a homotrimer, and this is a tetramer. The neuraminidase is shown in blown up here. Now they're important surface proteins because the hemagglutinin is the viral attachment protein. So it binds sialic acid residues on our cell surfaces and basically opens the door to our cells. So the virus can get in and begin to take over the cell and turn it into a virus factory. And so our primary target against influenza viruses in vaccines is the hemagglutinin. So some vaccines solely express the hemagglutinin protein, and others have the hemagglutinin, some neuraminidase, as well as some of the other proteins of the virus. Now, the hemagglutinin, I won't dive too deeply into the antigenic sites, but it's a large molecule, and our antibodies target different parts of it. And so those are highlighted in light blue here, where there's 5 antigenic sites that really are potent neutralizing antibodies will bind to those sites and block the virus from binding to cells, or the second function, which is fusing the viral envelope, which is shown here, to the cell. And then the genome is made of 8 segments of negative strand RNA, and this enables reassortment. So this segmented genome is somewhat unusual in the virology community, but in the virus world, but there are other segmented viruses. And this gives it tremendous evolutionary power where 2 viruses can co-infect a person or a pig or a chicken, and then when they infect the same cell, reassort, and you have 256 combinations. So these are can be quite different genotypes that come out. And not only can they do that, but the RNA-dependent RNA polymerase has a very high error rate. And so because of that error rate, influenza viruses are constantly changing. This requires the continuous comprehensive virus surveillance and necessitates frequent updates to the vaccine. So the replication of influenza is so error prone that it kind of is said to live on the edge of error catastrophe. And that could be a disadvantage for the virus because it's close to the threshold of extinction. Many defective viruses or non-infectious viruses are created during replication. And just to give you a sense, it makes so many errors that on average, every influenza virus particle has at least one nucleotide difference from every other influenza virus particle. But this can be a huge advantage for the viruses. The increased adaptability, the variants that are created every rounds of replication, that they're selected upon any evolutionary pressure, and it's a huge evolutionary benefit in invading host immunity, which is really why we have to update the vaccine all the time. And the other point I want to make about flu viruses is that they survive as a population of viruses, not just as a single virus. And so to drive home this point about existing as a population of variants, say you have a naive child that gets infected and they have an explosion of influenza in their upper respiratory tract. And so the virus replicates tremendously. You get many, many millions of particles created over the course of infections. And you could have an initial population that has all these point mutations, you know, from A to U listed on this slide I stole from Esteban Domingo's review here. But then if you put that under some selective pressure, say immune antibodies that would neutralize on the HA, you now select for a subset of viruses that could escape that pressure. And some of those subsets may share common point mutations across that subset and then have other point mutations thrown within. And that, so for example, the most fit virus group might be this top group with a large relative size, but then there's another one here and another one here. And when this person breathes out, coughs, sneezes, and another person inhales that also has a similar immune pressure, they further select these viruses. And so this is going through our population. So it's a population of viruses going through a population of people with different immune histories. And to give you a real world example of the dynamics of the H3HA gene, this is a long time ago now, but it really paints the picture. We had 3 groups, major groups circulating genetically, the 3C2A, the 3C2A1s, and the 3C3As. And this is October to January. And what you can see is the proportion of viruses in this group changing just in that time frame and selecting for this virus that was in this group, but also had 2 additional point mutations, 121 and 144. And then in the 2A1 group, we had a pretty widespread here, and it continued to stay pretty diverse throughout that season. And then the 3C3A group, you can kind of see the transition from this high level of viruses with the K192R to a bunch of viruses with the 197. And so this is really just trying to illustrate all these different clades were co-circulating in our population at that time. And it gets very confusing if we compile all those pie charts together into one series, but it gives you an idea of how many different flu viruses are actually infecting people in any one time. And so now we'll get into the antigen selection part. What makes up vaccine selection. So it's a complex biological system, and we have to do it 6 months in advance. As I just indicated, there's a swarm of variants circulating in our population. We also have host genetics that can impact how well the virus grows in different people, and also our prior exposure to influenza viruses, prior vaccine history drives immunity in different people differently. So how do we deal with all this? There's some key questions that are addressed as part of vaccine antigen recommendations. And the goal is really to identify antigens that elicit immunity to protect against diverging viruses that will likely co-circulate in the future. One of the biggest misnomers, I think, about influenza vaccines is that you're trying to guess, people will use the term guess, which is not accurate, what will circulate in the future, like there'll only be one thing. And really what we know about influenza viruses, and I just showed you, is what we know for sure, there'll be many viruses that co-circulate in the future in each of the 4 major groups. And so what we want to really do is identify antigens when we use them to immunize things that protect against a diverse group of viruses and will protect against the majority of viruses or will protect the important risk groups that Dr. Brion mentioned right at the outset, because that may be the most important group to protect. And so what are the genetic, where are the epidemics happening? What are the genetic subclades that have emerged in our population, and are they associated with those epidemics? Are new emerging variants spreading geographically? Are the emerging variant viruses antigenically distinct from contemporary viruses? And so that word antigenically, I'll use it a lot. And what we really mean by that is when we have immunity to a certain group of viruses, does another group of virus not get blocked by that same level of antibody, polyclonal antibody sera? And so that's how we call that antigenic drift, when it drifts away. And I'll use that term a lot. What is the proportion of new groups and what groups is or are likely to predominate? And notice groups is there. And do current vaccines induce antibodies in humans that protect against co-circulating variants? So antigenic distinction is often we use a naive animal model for, and the current vaccine antibodies in humans we use post-vaccination serum for. And then is the current antigen likely to provide the best protection, or is a new prototype needed? So of course, that's the key question. And one of the other big factors is, if a new prototype is needed, do you have the data for that new prototype that illustrates it'll work against not only the group that you're concerned about, but also other viruses? And so here's some of the key data used to address those questions. It's epidemiologic data, it's virus surveillance, it's genomic characterization, antigenic characterization, data integration. So this is a lot of computational biology, post-vaccination human serology, as I already mentioned, vaccine effectiveness studies, and I won't show details about vaccine effectiveness studies today, but this is a global consortium. It's really looking for VE that's lower than expected. And unfortunately, just in the cadence of way flu vaccine works, we have to be very proactive. So often vaccine effectiveness studies have to occur after a vaccine has been selected, but they are a good check of whether or not that selection was optimal. And then the availability and characteristics of a new candidate vaccine antigen. And so surveillance, everything begins with strong surveillance. And luckily in flu, we have the Global Influenza Surveillance and Response System, also known as GISRIS, which is critical to vaccine antigen updates, as well as pandemic preparedness and understanding emergence of antiviral resistance and all the other things we care about in public health. But I'm going to talk about it from a vaccine perspective today. There's 147 WHO national influenza centers. These are the front lines in each of the regions that we deal with, but of course they work with frontline individuals in each of their member states, which there are 123. They test 50 to 150,000 flu respiratory specimens a year to identify positives. They share representative specimens throughout the year with the WHO collaborating centers, or CCs, which there are 7, 5 that focus on human influenza, and 3 that work on animal influenza. Some work on both. And they are circled here. So the NICs are the blue diamonds in this map, and the CCs are the red and orange circles. Then we have the 4 essential regulatory laboratories. An example is like FDA or MHRA in the UK, and then 12 H5 reference labs. So nowadays, really what we've converted to is using genotype to phenotype analysis, which is central to vaccine antigen selection. And so this starts with phylogenies of all the viruses that are circulating. This is a nice source where all the data that's put into GISAID and other public repositories, but the global influenza database, GISAID, and can be visualized very easily using NextStrain, which is here, or NextFlu. And what we do is identify emerging lineages early. The NICs are regularly sharing samples, and some are sequencing themselves. And then we ask the question, which clades are co-circulating? And so, as I pointed out, just in this example from 2022, you have multiple clades of viruses that are always co-circulating. And then the question is, are they disseminating globally, or is it regional by a country? And so here you can see that in just the color coding. You don't have to know which clade is which, but you can see some large countries in area like the US, even on one coast, will have a different virus than another coast. And in other parts of the world, you'll have some very different viruses that are circulating. And so some of the things we do is look for the movement of viruses from one geographic region to another, and then displacement of those pre-existing viruses by that new virus that has come in. And then another key question is, are they antigenically distinct? And so here we have a phylogenetic tree on the left here. It's very small. You're not meant to know, it's not pretty like the next clade tree, but we have all the key amino acid changes. And then there's a few virus names here that are representative. And so this is in a case where we're looking at virus antisera against, for example, Darwin 6 2021, which was in our vaccine last year. In the Northern Hemisphere. And you can see it's in the tips of the trees where they're tested are actually color coded. So all those dots that are yellow are viruses that were tested, and they have less than a 4-fold reduction against the homologous titer. Then over here, we have a panel of antisera against different viruses. And so Tasmania 503 represents the Cambodia-like recommendation from the 2A1 group that was made previously. And you can see that is showing reductions against all these viruses, which are in clade 2A2. And so they have some key amino acid changes, such as this 159 and 160 that's bolded here. And that's just a few amino acid changes are driving that resolution that ferrets give you, because naive ferret model gives you very high resolution of antigenic changes. But we can also interrogate new potential candidates, such as Maryland, which is now at the top of this lineage and really expanded in North America last year. But when we test all the viruses across the phylogeny here with that Maryland antisera, you can see those at the bottom of the phylogeny have reductions. So when you compare Maryland to Darwin 6, you say, well, Darwin 6 is better, right? And then this is Darwin 9 egg. So we also use the egg-based antigen in our tests. And then one of the keys to how well they work in humans is really how humans respond to the vaccine. And so this is some data that helped drive the change to a Darwin 6 from the previously circulating or previous vaccine virus, Cambodia E0826360. And it's a high-level view looking at how well the different emerging groups at that time evade vaccine-induced immunity in the human population. We have panels that span from 0 to 6 to 35 months, all the way to older than 64, as you go down in these rows. And so we segregate them by age, because sometimes we see age-specific escape or age-specific neutralization. And here we're looking at testing viruses that are emerging or have previously existed. So 1A is an older cladovirus, but it had developed new emerging groups. It was represented by this Togo 771. 1B kind of represented an older virus, Hong Kong 45, that was actually the vaccine prior to Cambodia. And so what you can see is blue is good. And as you get into the light orange, that's where the lower bound of the 90% confidence interval of the range for the geometric mean titer difference is touching the 50% mark. So that's not very much reduction. So it's not like ferrets. In humans, you get a more subtle reduction, 2 to 4-fold reductions. And so really you have to do many sera and use statistical analysis, or at least compile all that serum together to see a real difference. And I'll dive into some of the data in a minute. But what you can really appreciate here is almost all the serum panels have some orange in them, and these are all 2A2 viruses like the Darwin 6, the Maryland 2, and the Alaska 01, which each have additional substitutions in addition to the Darwin 6. So we'll dive into that data a little bit. So this is now looking at the individual responses, and I blew it up just so you see two of the pediatric population panels, the 3 to 8 and 9 to 17. And we can focus in on almost any one of them, but we'll just start at the top row here. So this is Flucelvax or inactivated vaccine 4. So it's egg-based and cell-based vaccines. And what you can appreciate is, we're getting a pretty good response to the vaccine. All the prior pre-vaccine sera is showing low geometric mean titers of around 30 in this row here. And it bumps up to 171, with 75% of those individuals having a titer greater than 40. And so a titer of 40 has been a correlate of protection from influenza virus. And then if we use the egg antigen, we also see a rise. And then when we use the Togo 771, which is a different clade and has undergone further evolution, you also see a rise. And so this is showing you that it is boosting against antigenic variants, but how well that boost is something we look at. And so 166 isn't very different than 171, for example. And then if we look at Hong Kong 45, which is an older virus, you actually see the geometric mean titer higher than the immunogen. And so this is in part because of immune memory. So when there's conserved parts of the hemagglutinin that when they're vaccinated with, you get a nice response. And so that's this immune memory, and sometimes called a back boost. But you can see all that forward boosting within these other clades. And then we get into the clade of interest that's emerging rapidly at the time, the 2A2 viruses. And you can see even against that antigenically distinct virus, very antigenically distinct by ferrets, as you remember, it had a pre-existing titer of 17. It goes up to 89, and 70% of the people have a greater than 40 titer. And so this is why getting vaccinated, even when you have a new antigenic variant, is helpful. And then this is same is true for some of these more advanced viruses. And so that's really you can think about that like a forward boost. And so then comes data integration, fitness forecasting. And so how fit is this new lineage that we're looking at, or the various new lineages compared to all the other lineages that are co-circulating? And then identifying the optimal vaccine antigen. And so I kind of showed you some data on this, but which virus antigens induce protection from the emerging groups? So we compile a lot of data across all the CCs that is shared. And you can look at this through antigenic cartography, such as shown here, where you see groups of viruses like that Darwin 6 group, the 2A2 viruses up here, and the 2A1 viruses down here. And each of these squares representing a twofold difference with post-infection ferret antisera. We look at the molecular structures and where the changes are occurring in the antigenic sites that I pointed out earlier, how important they are. And here's some fitness forecasting, really illustrating that some clades will likely die out, but those are the ones already dying, and there will be multiple clades that co-circulate, and probably their progeny will evolve. And then finally, you'll look at the candidate vaccine viruses that you have available and antisera to those as to how well they work. So here I have an HI test from our collaborating center colleagues at Vidril in Melbourne, Australia. And here it's kind of a nice panel. It's mostly vaccine antisera, but you can see viruses that were circulating at the time. This was really looking into a potential Southern Hemisphere 2022 change to Darwin 6. You can see the older vaccine did poorly. The red is bad. The vaccine being used, the Northern Hemisphere 21-22 vaccine, did better. And then if we change to this Darwin 6, this is a cell-based, and here's the egg-based over here. You see good protection of all these 2A2 viruses, but not very good protection of the 2A1, the 3C2A1B 1A viruses, and reduced against the 2A1 viruses. The 2A1 and 2A2 are genetically very closely related. Okay, so to summarize, influenza viruses rapidly evolve and evade immunity generated from prior infection vaccines. There's many divergent populations that are co-circulating, and the virus evolves rapidly to escape host immunity. One of the major goals in vaccine antigen selection is identifying those antigens that elicit immunity against diverging viruses that will likely co-circulate in the future. And we have to use many data sources to address the key questions, which are kind of restated here, but identification of new lineages, dissemination, antigenic characterization, immune escape from human sera, and the risk they pose to specific groups in our population. And then the genotype to phenotype approach is really now central to all of this activity, and integration of that data is central to how vaccine antigens are being recommended. And there's always many efforts underway to overcome the contemporary vaccine challenges and improve antigen selection. We're having continuing use of next generation sequencing across the globe at many of the national influenza centers. So this decreases turnaround time from swab collection to sequence deposition in public databases, which is very important. A lot more data integration and fitness forecasting and those complementary approaches. And a lot of the CCs now generate many candidate vaccine viruses early, and we down select as the vaccine recommendation date approaches. I can't end without acknowledging all the people that play such a huge role in this, the GISRIS and all of its parts, including the other WHO collaborating centers and essential regulatory labs, US partners, the data integration folks, and CDC staff. And if the slides are shared, I have many acknowledgements within my own team. Thank you very much.
Thank you so much, Dave, for presenting this huge amount of work. This amount of work was done within 4 days with a room full of experts reviewing this amount of data. And this time we had the chance printing out the data package. It's like 10 centimeter or more than 10 centimeter, this type of folder data. Thank you so much, Dave, for this. We will take questions after the second presentation. So if you have questions, please type that into the Q&A, and we will address that after the second presentation by Dr. Richard Webby, who is the director of the WHO Collaborating Center in St. Jude Children's Hospital. And he will be talking on the avian influenza, including H5 and the candidate vaccine viruses. Richard?
Great, thank you, Wenqing, and good morning, good afternoon, good evening all. So as Wenqing mentioned, we sort of sit together for 4 days to discuss vaccine virus selection. Typically, the first half day is spent, of that meeting is spent discussing zoonotic influenza. And that's what I'm going to do, is a very high level overview. Dave's sort of done the hard work of describing a lot of the nuts and bolts of what goes into these decisions. I'm going to give you a little bit of a flavor for how some of those same considerations are used in terms of pandemic preparedness and readiness against some of these other influenza virus threats that are in animal populations, but not yet in humans. And sort of this is, again, the 30,000 view of what influenza really looks like. We know that it's these animal populations of the world, particularly some of the waterfowl, that harbor a huge diversity of influenza viruses. So all different flavors and shapes of flu viruses circulate in these populations. And for the most part, they stay within those populations. There are some viruses that move between these animal populations, and every now and again, they sort of try their luck in the human host. So we have a number of zoonotic infections, so transmissions, direct transmissions from these animal hosts into a human. Luckily, the vast majority of those are self-limiting, so there'll be one, on occasions, maybe a small number of human-to-human transmissions, but those viruses don't take off. And of course, when they do, that's when we have a pandemic, and that's what we are trying to do with what I'm going to talk about for the next 15 minutes or so here, is what can we do for preparedness when these viruses decide one day they don't want to be a chicken virus anymore, they want to become a human virus. And if they start spreading, what happens? And these viruses are trying this all the time. And so this is just a summary of the number of zoonotic infections over the past couple of years or so that have been detected across the globe. And again, we don't need to go into too many details, but you can see that it happens around the globe. It happens with different subtypes of viruses. Some of these on here are viruses from birds that have infected people. Some of these are viruses from swine that have crossed over to infected people. But again, these viruses, the main take-home point is they continue to try. We continue to get these zoonotic infections, but certainly in this case, the vast majority of these, if not all of them, have no human-to-human transmission. So animal to human, but no human to human. Yeah, of course, if that changed and these viruses do start to go human to human, what would happen? And certainly from a WHO perspective, vaccine development would be the first thing that would happen. And so we've all seen versions of this slide that sort of go through how long that would take. So from identification of a virus that a new virus that's starting to spread amongst humans, so not the H1s or the H3s that Dave talked about, but perhaps an H5, perhaps an H7, or maybe a different flavor of H1 or H3. And it's about a 5 to 6 month process. Much of this can't actually occur until vaccines are identified, the pandemic virus is made, certainly with our current vaccine technologies, which are fairly specific for the immunizing strain. Again, as Dave said, lots of effort going in to make more broadly reactive vaccines, but this is what we've got to work with at the moment. And if we think about this, what can we do actually now in times of peace? Again, when these viruses are trying this zoonotic infections, but don't yet have the capacity, there's one thing that we can be doing, and that is taking this first step out of a response timeline. And so this is seed strain preparation and verification. So this takes about, depending on virus, in the 4 to 6 week range. When we have identified a virus, what actually goes to manufacturers, at least for the inactivated or live attenuated vaccines, are I guess manipulated viruses. So they have to be, if it's an H5, for example, which are highly pathogenic strains, have to be manipulated to reduce their virulence before they can be safely handled en masse. These strains have to be tested. And so the idea, but for why GISRA spends effort in making these seed strains is, if these viruses do take off, then we're 6 weeks ahead of the game, and we can take 6 weeks off the vaccine response. And of course, 6 weeks off a vaccine response could save a lot of lives. So just looking at what has been done, this is a table. So every February and September, when we meet to discuss the vaccine composition, there's a summary produced and posted on zoonotic activity. And in that is a list of what, in this case, just H5 vaccine candidate virus have already been produced over the past 20 odd years. Again, the detail's not important here, but you can see there is a rather large list of viruses here just for H5. And these have been developed really to try and cover the antigenic diversity of viruses that have been seen and sort of more recently being seen. It's been driven, how do we select which ones we actually make? A lot of it's driven by zoonotic infections. So there is this huge amount of diversity in particularly the waterfowl populations of the world. We don't have the resources to make candidate vaccine viruses against all of those. So we tend to, the selection process is driven by viruses where we have seen zoonotic infection. So those that are perceived of most risk. And again, as I said, the purpose is to cover the major risk groups of zoonotic viruses, and really to shave that 5 to 6 weeks off the vaccine response. But these are also very valuable reagents, both in terms of scientific reagents, manufacturers to get experience handling some of these viruses. So they have a lot of uses other than just vaccine manufacture. And this list gets longer as new antigenic groups of viruses emerge. How do we determine that? And this list I actually stole from Dave's slides. So he just presented this in terms of what data is used to decide, well, out of all of this swarm of viruses circulating, which ones do we want to make a candidate vaccine virus against? For the seasonal, it's based on all of these. If we look at that zoonotic, you can see it's fairly similar. So certainly epidemiologic clinical data, virus surveillance, keeping in mind that a lot of these viruses we're looking at here are not yet in human populations, so they're circulating in animal populations. So over the years, we've developed some very, very strong partnerships with institutions and individuals in the veterinary sector as well, which are key partners in these efforts. The genetic characterization, lots of sequencing, lots of antigenic characterization, the same. Post-vaccination human serology, sort of a cornerstone of seasonal decisions. Of course, a lot of the viruses that we're talking about in the zoonotic space, humans have no pre-existing immunity to it, and we know that. So this is an area where this is far more heavily used in the seasonal vaccine strain selection, not so much in zoonotic, unless we're talking about some of these swine viruses, which have a lot more antigenic similarity to viruses circulating in humans. We don't have vaccine effectiveness data to help us pick zoonotic strains, and the availability characteristics of strain is also used. So overall, very, very similar data sources that go into a zoonotic decision as a seasonal decision. But typically we do have a lot less data than what sort of Dave showed. So I want to very, very quickly run through one example of this in terms of our thinking and how this works. So last week we were in Geneva discussing this, and some of these slides were presented by Todd Davis at the CDC at the information meeting. So H5 from the September to February period, which is from the last time we met to make vaccine recommendations to this most recent time, each of these dots represents a reported detection of a highly pathogenic H5 virus. Most of these, the so-called 2344B virus, which has been responsible for this resurgence in H5 activity globally over the past couple of years. You can see lots of activity through Europe. There's not as many dots as you might expect in Southeast Asia or Southern Asia, but some of these countries are endemic and don't necessarily report every outbreak. And of course, a little bit new for these viruses, North America, and in this reporting period, the first time these viruses were detected in Central and South America. These viruses weren't just circulating in birds, they had caused human infections. So these were just a line listing of the human cases that were reported in the period from September 2022 to February 2023. So again, these viruses keep on infecting humans, and each of these cases, no evidence of human-to-human spread. Genetically, this is just a subsampling of the tree, and this is clustering on these 2344B viruses. As they've spread globally, they're starting to settle now into some really geographically defined genetic clusters. Either the Americas through Europe, Asia, and Africa. If you can have a look on this tree, the red ones here are candidate vaccine viruses that have been recommended and developed in the past. And as you can see, the further these viruses move away from the candidate vaccine viruses, that signals to us that maybe we need to be thinking about development of additional CVVs, perhaps to represent some of these emerging groups. And again, we do the same hemagglutination inhibition assay that Dave showed. And again, this is just a representation. If we home in on this particular group of viruses here, again, so these are just representatives of, this is a US CDC HI assay using representative viruses from the US here. The homologous titer for the current vaccine virus here is 320, and as the numbers get smaller and move away from that, again, it's suggesting that not only are these viruses moving away genetically, they're also moving away antigenically. And of course, antigenicity is the key to potential vaccine effectiveness. So in this case, due to this genetic movement away, due to this antigenic movement away from the available list we have, we made the decision that we should probably go ahead and add a new CVV to this list, just in case a virus that's more like some of these, and this virus here is more like some of these, just in case that's the virus that makes that leap, makes those key changes and starts to spread. So to summarize, Zoonotic CV development has been an integral part of vaccine composition meetings since the early 2000s. And again, as I've said multiple times, the purpose is really to cut 4 to 6 weeks off that vaccine response. So these are pandemic preparedness activities. The process and data needs are very, very similar to the seasonal decisions. And that makes a lot of sense. The seasonal decisions are based on decades of experience and information and knowledge. And so we're using that to help us inform decisions in the zoonotic space as well. I think one of the highlights for me of this effort is really the strong partnership. Oops, I've got an extra L in here, sorry. But OFLU, so the animal expert panel of the OIE and FAO are now integral parts of these decisions. They bring a lot of information, they bring a lot of expertise to these discussions. And the take home on everyone to take with them is the zoonotic threat from influenza is not subsiding. We're seeing a large amount of detections of viruses, avian influenza viruses in humans. We're seeing many of them infect, well, not many, different types of these animal influenza viruses infecting humans. Luckily, most of those don't have the capacity to spread human to human, but one day that's going to change with one another. So again, thank everybody involved in these, and Wenqing, I'll stop right there.
Wenqing, I don't hear you.
Now?
Yes.
Okay, thank you, Dave. Thank you so much, Richard, for controlling the time so well. I've seen many names that I'm familiar with. So you are all from the WHO's GISRA system, all the partners associated with the WHO's GISRA system. Welcome again to you on board. I would also like to introduce another panelist who will join me to addressing questions. Dr. Aspen Hamot also joined, and the panel will be able to address the questions from the floor here. So if you have questions, please type into the Q&A. If we don't have enough time to address all the questions in this webinar, we'll be able to answer you in writing. So with this, let's kick off. Because this webinar is focusing about vaccine composition. So let's start with actually 2 questions about universal vaccines. So the question is the current progress about universal vaccines in the context of the CVV selection. So Dave, would you be able
I can give it a start, and I think it also pertains to zoonotic vaccines as well. So universal vaccines are just a fantastic goal for influenza. And I think most people on the call probably know what that means, but really the initial definition was something to the effect of vaccines that would protect against at a 70% level. So that's a very high bar against all different strains of viruses that would circulate in humans. So that's a very challenging, as I showed you, just for doing strain specific vaccines, it's a big challenge. And so this is something I don't work on. This is not my area, so I'll just say that right away. There's many studies and work supported across the globe in this setting, and a lot of them target specifically conserved regions of the flu virus. So, for example, the HA stalk, Dr. Florian Kramer's group and Peter Palazzi's group at Mount Sinai have done a lot of work in that space. This is an area that there is less evolutionary change because that part of the hemagglutinin is actually a molecular machine that has to unfold and fuse with the host cell membrane. So it has less room for viral evolution. But I think the challenge there is producing really high titer against that stalk, so high affinity antibodies against that stalk. It's kind of a boring looking protein, I think, to the immune system, and probably designed that way by evolution of the virus. Others go and target the ectodomain of the matrix protein, which is, again, very conserved part, but it's very short. You couldn't even see it on that virus particle that I showed you, and kind of covered up by all the hemagglutinin and neuraminidase molecules. And so it may be tough for antibodies to get into. And so I think there's a tremendous amount of research going on there, and there is progress happening there. We're learning a ton about the immune system, but I think also another way to think about universal may be different than those highly conserved regions, and that may be using multiple approaches simultaneously. Different platforms, different vaccine strategies could also potentially help create a universal immune response. And I'm not sure as much work is going on there. But that's where I'll leave it.
Thank you, Dave. I don't know if Richard, do you want to add anything from zoonotic flu perspective?
No, well, I'll add maybe a couple, because I think actually the zoonotic space or the pandemic preparedness space is an area where perhaps these more universal approaches actually have a home. I think, sort of as Dave alluded to, a lot of these more broadly reactive immune responses aren't quite as protective. So for a seasonal vaccine where you really want to protect as much as you can from even mild disease, maybe from transmission, in the zoonotic space, when perhaps there's an emerging pandemic, I think you can lower the bar of expectation for a vaccine to at least stop severe disease and death. And so, I think there's a lot of utility for more universal approaches within the zoonotic area as well.
Thank you both. And I would like to add that WHO also is working with research academia. There's a vaccine roadmap in place. So although it is very challenging to jump into the universal vaccines, but stepwise, for example, vaccines with broad spectrum of protection are being focused and developed as stepwise approach. Let's move on to the next question about correlates. I think this again is the question related to this webinar topic, correlates or protection. Would Dave, would you give some comments on that?
Sure. I kind of mentioned it, and the one main correlate of protection that influenza has had, it's kind of a long-standing correlate from some older studies, was titers greater than 40 were neutralizing titers or inhibition titers greater than 40 were considered protective, but really this was protective of about half of the individuals. So 40 is a very low number. I think, you really, if we're talking protection from infection, we want higher titers like 80 and 160. And this is one of the challenges with flu vaccines. Sometimes it's very difficult to get high titer, even 4 weeks after vaccination.
Great, thank you. Richard, if you want to add anything, just unmute yourself. Yeah, otherwise we'll move on. Let's move on to the surveillance question then. One question is about the viruses coming in from countries. There's relatively low number of viruses coming in from Africa. What is the implication to the virus selection? Dave?
I can try to take this. I think that's a fantastic question. We are so concerned about bias in the selection process. We continually try to work to reduce bias. And so that bias could be a numbers game, few viruses from a country with a large population and actually a pretty good flu season, like 20% positivity or something like that. And so there's a lot of activity going on to try to weight the data a little differently, or at least look at it differently, and try to understand, well, it could be that one virus that you're getting from, or these 10 viruses that you're getting from this country or region really represent 10,000 viruses from a country that has tons of viruses that are being sequenced or looked at. And so that's a huge problem. We do need all the NICs, and we need more NICs, and we need everyone to be sending viruses in regularly or sequencing viruses regularly, and that will even the playing field and likely improve vaccine selections, and definitely improve wherever that region is that's underreporting or underrepresented. It'll improve their representativeness for vaccine selection. So there's a bit of a win-win in doing more at each region or in each country or territory.
Indeed. Thanks, Dave. And just to highlight two keywords in the influenza community, one is a timeliness, and the other is representativeness. These are the two key words in the flu community. Okay, let's move on to the next question, specific to Richard, about to identify potential risks of zoonotic transmission of avian influenza, whether a strategy aiming at routinely sampling people exposed to poultry or wild birds would be interesting.
Yeah, so again, another great question. So, you know, again, we struggle a little bit in terms of the sensitivity of our surveillance in particularly the animal world. So the discussion we just had about representativeness of samples for a human selection, it's you know, it's worse, I think, for a zoonotic. So there has been discussion about, you know, maybe we're better off focusing the surveillance effort actually at the people exposed to those animals. And these studies are going on, you know, and when information is available from those studies, we do use it. I think some of the problem is, you know, luckily these are relatively rare events. So, you know, even though we're probably only detecting a fraction of what's actually happening, they're still rare events. So setting up these really expensive studies or surveillance efforts to particularly focus on these concentrations, you know, unfortunately, many times don't actually detect the events crossing over. So I think they are important. They are important in terms of a holistic approach to surveilling. But again, it's a little bit of a needle in a haystack in terms of understanding.
Thank you very much, Richard. Let's move on to the next question. Actually, maybe I can try to address both questions. One is from the email, and the other is a question about the criteria of licensing pandemic influenza vaccines. Well, basically about neuraminidase. Well, whether we are looking into neuraminidase titers in the vaccines.
So the licensing question I can't address, but the neuraminidase we do look at. I didn't present it because it's not the primary target of all vaccines, and it's not the most neutralizing target, but it's something we consider heavily. We do a lot of genomic analysis where we look at reassortment. So there's a lot of intra-subtypic reassortment. So many people think about reassortment of influenza virus like an H5 reassorts with a human virus, and they're very different outcomes, but you can have two very similar viruses reassort, and then you see the neuraminidase and HA changing partners, and there's a really important dynamic balance between the hemagglutinin and the neuraminidase. And so we use the neuraminidase analysis both as a major part of fitness forecasting, but we're also doing a more limited antigenic analysis of the neuraminidase. So we take that same type of antisera that I showed you, and we do neuraminidase inhibition assays using ELA, which is the target of the neuraminidase. And so again, that's done less, but it is part of the vaccine strain selection process. And we do try to have neuraminidase that are like the HA, that are representative of the viruses that are circulating.
Thank you very much. Yes, Richard, please go
Maybe
ahead.
just to add a little bit, again, I'm as much as I try and tell my children I know everything, that's of course not true. So yeah, and I'm not so familiar with the regulatory aspects of this as well, but I suspect they change a little bit from region to region, but it is based on that, a lot of it based on that correlative protection Dave mentioned before of the HIF40. But I will just mention that within just within these discussions, the vaccine composition meetings, we do have representatives from the essential regulatory laboratories who do know this side of it. So these people are in, and perhaps next time we can have them join something like this.
Thank you, Richard. Thank you, Dave. So let's move on to the next question. A participant is interested in adjuvants. So the question is whether it's necessary to have a library of adjuvants to prepare for outbreak, and whether immune informatics is a useful tool for the prediction of antigenic drift and shifts. Adjuvants.
Maybe going to give a little crack at the adjuvant, and I think because it's, you know, although it's used in, and again in both sides of this equation, but I think it's particularly important in some of these vaccines, again, using the traditional vaccine approaches for some of these avian antigens. So the H5, the H7, the clinical trials that have been conducted in that space, typically the immune response is not robust to those antigens. And so adjuvants, which will boost those immune responses, are probably critical to vaccine responses to emerging events, particularly if you want to give a single dose. So, you know, I know there are efforts. I think there was another question about the stockpiling of those adjuvants. You know, there are efforts to go into that as well. Yeah, I must admit, I don't quite know sort of the global scope or manufacturing capacity of adjuvants, but clearly for a pandemic vaccine, they are very much in the discussion and very much of a lot of countries' plans.
Yeah, I agree with everything Richard said, and I think there's plenty of space for safe and effective adjuvants to be developed. I think that is a huge thing with influenza, and would go a long ways towards all the things we're trying to do, particularly, you know, in boosting the immune response, even to seasonal viruses. All that I showed you, that was the back, the memory response and the forward response. Really, the higher, tighter you go, the more you'll see that. And so that is towards the universal vaccine. And I think they're likely very important. And when we're trying to target very maybe not immunologically robust proteins or parts of the proteins as well. So I really think it's a great space to work in and develop more.
Thank you both. Superior, please remind me, the webinar is for 1 hour or for 1.5 hours?
Hi, Wenqing. It is for 1 hour.
Okay.
Nevertheless, we can run over if it's okay with the participants. I think it's such an interesting discussion. I think if it's okay with everyone, we could run over a little.
So maybe we can take one more question, because this question I think is very important. It's asking about mRNA flu vaccines, well, in relation to the vaccine virus selection. I think maybe Dave, you can start, and then others can add.
Yeah, so what's the question specifically about mRNA flu vaccine? I'm sorry, I haven't been tracking all the questions as well.
Vaccine virus recommendation for mRNA vaccine.
Oh, yeah. So that's a great question. I mean, initially, most products will use the recommendations that we're developing now as part of their portfolio that they submit to regulators to illustrate that it works well against that. I think there was also a question about, oh, here, as the field moves forward, the vaccine recommendation may not need to be restricted to a single strain per subtype, and even consider, other than the HA, a correlation of protection. So how will that work? And so we don't know the answer to that yet. There has been discussions internally with the VCM committee members to try to understand how that will work. There isn't a licensed product yet, so that's you know, why we don't have a system for it. But I think one of the keys will be the goal that I mentioned, which is how well does that particular antigen, or if there are multiple mRNAs, for example, against H3, how well does that protect not only against maybe the recommended candidate, but against other viruses that are also co-circulating. So anything that would broaden that, I think, would be easily a strong recommendation. So I think that's the way it will go. We don't have the defined guidelines for that as a group yet. But we're hopeful.
Indeed. Thanks, Dave. So, of course, WHO will be monitoring and tracking the progress of the new product development that certainly would improve the pandemic influenza response capacity on that. So with this, I think we need to wrap up. As I said, this is one of a series of webinars. We'll continue organizing such webinars and focusing on specific subjects. For the questions not yet answered, we'll answer in writing. So please don't worry. So taking this opportunity, I would like to thank our two presenters, David Wentworth, Dave, and Webby, Richard, and also my another panelist, husband, my colleague in the panel. So thank you so much to you all. With this, I hand back to Superior.
Thank you very much, Wenqing, and thank you for excellent moderation as well. I'd also like to thank my colleagues who've been working on this in the background, Dr. Yubika Latinovic, Dr. Priya Umachandran, and Jiangfang Liu for all their support in making this happen, from the technical aspect to all other aspects. And of course, Dr. Aspen Hammond, who's been responding to some of the questions, who works on influenza with Wenqing. And last but not least, thank you to all the wonderful participants. I've seen participants from the Far East, from Japan, all the way to countries like Bolivia. So I hope you found it useful. We look forward to your feedback, and we look forward to you joining us in our next EPI-WIN webinar. The next one will be on 15th March, and that will be on cholera. We will also have another influenza webinar towards the end of the month, or we will get back to you soon with the dates. Thank you so much. And thank you to Richard and David once again. And also to the lady whose vision it has been doing these webinars, Dr. Sylvie Briand, our director. Bye, and see you next time.
Bye all.
Bye all. Thanks very much.